5, 4, 3, 2, 1. So,
Good evening to ladies and gentlemen in the United States, and good morning to ladies and gentlemen in Taiwan. I'm Lotus Wang, your MC for today. Today, we are very delighted by your presence to the 2021 NCI, TVGH, and NYCU Joint Symposium, and the opening ceremony to Stephen J. Solarz International Laboratory. This conference is organized by the National Cancer Institute, Taipei Veterans General Hospital, and the National Yang Jiao Tong University. Before we started, I would like to express my sincere appreciation to all of you, especially our distinguished speakers from the USA and Taiwan, the AIT officers, and the officers from the Ministry of Foreign Affairs Taiwan for joining us on this beautiful day. So without further ado, it is now our great pleasure to invite Mrs. Nina Solarz from the Stephen Solarz Memorial Fund to deliver the opening remarks. Mrs. Nina Solarz is the wife of the late Congressman Stephen Solarz, for whom the International Laboratory has been named. She led the efforts to create and fund the laboratory when she and her family created the Stephen Solarz Memorial Fund at the Foundation for the National Institutes for Health in the United States. Now let us welcome her to deliver the opening remarks. Mrs. Solarz, please.
I am honored to be introducing the symposium and ceremony for the opening of the Stephen Solarz International Laboratory in Taiwan. Named in memory of my late husband, Congressman Stephen Solarz, to this distinguished group of scientists, government officials, and higher education officials gathered to inaugurate this unique joint U.S.-Taiwanese enterprise. After several years of negotiations and a highly competitive review, scientists at the National Cancer Institute of the National Institutes of Health in the United States and scientists at Veterans General Hospital and its affiliated teaching institution, National Yang Jiao Tong University in Taiwan, agreed to link their respective laboratories and cooperate on protocols utilizing stem cell, epigenetic, and immunotherapy regimens to target cancer cells and to establish exchange programs to train future leaders in cancer research. These agreements were approved by the United States Department of State, American Institute in Taiwan, and the Taiwanese government. This unique collaborative effort grew out of nearly 10 years of support by the Stephen Solarz Memorial Fund of the Foundation for NIH to the Thoracic Epigenetics Laboratory of Dr. David Schrump, Chief of Thoracic Surgery at NIH. The laboratory had already established the Stephen Solarz Fellowship Program to be awarded to foreign doctors and medical researchers, and this ultimately led to the collaboration between the lab at NCI and the hospital and university in Taiwan. After a Taiwanese doctor was chosen to receive a fellowship and worked in the NCI laboratory for 2 years, we were introduced to the sophisticated research protocols being developed in laboratories in Taiwan, led by 2 distinguished Taiwanese doctors and researchers, Dr. Muhua Yang, Provost of National Yang Jiao Tong University, and Dr. Shihua Chao, Chair of the Department of Medical Research at Taipei Veterans General Hospital. It was then that my family and I proposed the opening of a joint program between NIH and the Taiwanese University and Hospital, which is now being inaugurated today. One of the missions of the collaborative research is to determine distinct features underlying thoracic cancers in Asian versus Caucasian patients. As part of this effort, clinical trials directly emanating from work performed in the Stephen Solarz International Lab will open simultaneously at the NIH Clinical Center and Taipei Veterans General Hospital. An exchange program within the labs will foster the collaborative effort, promote protocol development, and enrich the training of students and postdoctoral associates. When my husband was chairman of the subcommittee on Asian and Pacific Affairs of the House Foreign Affairs Committee, he had worked closely with the Taiwanese people, most significantly to help establish an opposition political party. Naming a significant medical facility in his memory is a most enduring tribute to him and his commitment to the people of Taiwan and to those of many other nations of the world to which he dedicated his life's work. Thank you.
Thank you, Mrs. Solarz, for the great speech. Now let's welcome the President of National Health Research Institute of Taiwan, Professor Liang Gangi, to give us some remarks. Professor Liang, please.
Can you hear me okay? This is Solarz. I'm really thrilled and honored to be here to participate in this event, witnessing a historical event. Historical, as I believe this is probably the first time a distinguished research laboratory from NIH working with its counterpart to establish a joint laboratory in Taiwan, Taipei Veterans General Hospital specifically. So it's really truly remarkable. And we all know that it takes a long process, almost it's 5.5 years in its making, and it involves the State Department of the United States, TECRO, and the Department of Foreign Affairs. But I think that, and also it came with the COVID-19, so it took a long time. But I think that sometimes it's better to give us a better appreciation as to what it takes to see its fruition. As someone witnessed this historical event, I couldn't help but looking back, and there are several words, a phrase that come to my mind: passion and perseverance, mutual trust and respect. Under the leadership of Mrs. Solarz, I think she had demonstrated once again her passion to the people in Taiwan and her persistence in seeing it through. I think it's probably fair to say that without her efforts, we probably won't be here to celebrate. I hope you can allow me to reminisce a little bit. I believe there was in the August 2016, few months after the beginning of this interaction, I was sitting in the lounge of Taipei Airport, waiting to catch flight to the state of Maryland to see my family. While waiting, I received an email from Mrs. Solarz suggesting that because knowing that I'll be in the in the States, she suggests maybe I can have just get together with David and also Randy to get more acquaintance with each other. I responded immediately, definitely, only to realize that there was really a family event. So the only clothes I bought were the polo shirt. I didn't even have the shirt, or not to mention about the jacket. So once I arrived in the States, the first thing I did is go to the Columbia Mall to buy several pairs of shirts looking semi decent. And both David and Randy were so nice, knowing that I didn't have a car to spare. They drove all the way from Bethesda to Columbia Mall, having a dinner together. I believe they named the restaurant was PF Chang. We're sitting in the corner of the restaurant. We didn't say much about the research lung cancer because that was really not my own research area of interest. But instead, we're talking about, I'm trying to convince them that I think that the history and the high quality work of the researcher from both the National Yangming University at that time, also Taipei Veteran General Hospital, and why this will be a win-win situation to form a team together. Meantime, David shared with us again his vision as to how to make this collaboration a successful one. I hope that both David and Randy agree with me that at that time, really you had the sense of mutual trust and respect, and also believe that such a feeling is really prevail among all the participants during the process. So I think that there's I'm very why I mentioned about those words, passion, perseverance, and mutual trust and respect, because I believe that under the spirit, I think that a very solid foundation has been built for this international laboratory to excel. So my best wishes to the success of this joint adventure. I hope that before too long, we can have all of you from the overseas coming to Taiwan get together to celebrate about this historical event. Thank you very much.
Thank you, Professor Liang, for the warm speech. Now let's welcome the Chief of Thoracic Surgery Branch, National Cancer Institute, Dr. David Schramm, to give us some remarks. Dr. Schramm, please.
Thank you for the introduction, Lotus. I first met Steve and Nina Solarz in 2006 when Steve was referred to my clinic at the National Cancer Institute. I saw both of them frequently as Steve completed multimodality care and follow-up for esophageal cancer. Following his death in 2010, Nina and her family established the Stephen J. Solarz Memorial Fund at the foundation at the National Institutes of Health to support work that was being conducted in my laboratory. This fund enabled us to conduct a first-in-human cancer vaccine protocol, results of which were recently published. This was a positive trial, and it now has supported further evaluation of this lysate vaccine developed in our lab in conjunction with several immunomodulatory agents in patients with lung and esophageal cancers. The fund also enabled us to establish an international fellowship program. Nina had requested, as we initiated the program, that it be international in scope due to Steve's legacy in U.S. foreign policy. Our first fellow was from Istanbul, Turkey, and our second fellow was from Taipei Medical University. While we were in Taipei interviewing candidates for the fellowship, we had an opportunity to meet scores of people at top medical institutions. Nina became very impressed with the quality of medical care and cancer research being conducted in Taiwan and suggested the establishment of an international collaboration between my lab at the National Cancer Institute and an institution in Taiwan to foster exchange of ideas and to accelerate the development of novel therapies for thoracic cancers. As such, we made several return trips to begin formal negotiations with medical institutions and to interview additional candidates. These trips were arduous yet always fun. They always involved a group effort. We were up and in our first meeting, typically involving a formal presentation by myself before 8 a.m., followed by multiple additional meetings and interviews, a quick lunch, and additional presentations, meetings, and interviews at another hospital in the afternoon. This very full itinerary was interspersed with visits with political leaders and meetings with various pharmaceutical companies. During her afternoon interviews with the news media, Nina promoted her vision for the International Lab, while the rest of us did what we could to foster foreign relations. Among the various institutions that we visited, we were particularly impressed with National Yang Ming Chiao Tung University and Taipei Veterans General Hospitals. During our visits to these institutions, we met two outstanding physician scientists, Dr. Muhua Shi and Dr. Shi Hua Chu, and it became very clear that we had complementary and potentially synergistic scientific and clinical interests. Following lengthy negotiations and a competitive review process involving several top-tier institutions in Taipei, the Stephen J. Solarz International Research Program in Thoracic Oncology was approved by the U.S. Department of State American Institute in Taiwan and the Taipei Economic and Cultural Representative Office. As Ms. Solarz mentioned, the mission of this international research program is to characterize distinct features underlying thoracic cancers in Asian versus Caucasian patients, to conduct cooperative protocols utilizing epigenetic and immunotherapeutic regimens targeting cancer stem cells in these malignancies, and to train future leaders in thoracic oncology and translational cancer research through an integrated and vibrant exchange program. This international program, which includes the Stephen J. Solarz International Lab at Taipei Veterans General Hospital, as well as dedicated space within my laboratory at the National Cancer Institute, will focus on four major projects. The first project is to utilize induced pluripotent stem cells to develop personalized cancer therapies. The second project involves the evaluation of aerosolized chromatin remodeling agents for lung cancer therapy. We feel that this is a very unique opportunity for collaborative interactions among our institutions, as improved local delivery of these drugs will lessen systemic toxicities and perhaps prolong survival in patients, as it has done in preclinical studies. The third project involves the investigation of epigenomic mechanisms regulating metastasis and response to immune checkpoint inhibitors, and the examination of mechanisms of resistance to these immune checkpoint inhibitors in Asian versus Caucasian patients. The fourth project involves investigation of mechanisms of lung cancer progression mediated by nicotine and cigarette smoke. These efforts have potentially broad public health implications for first and second-hand smoke exposures, as well as vaping. In summary, the Stephen J. Solarz International Research Program in Thoracic Oncology provides a unique opportunity to foster international cooperation regarding the diagnosis and treatment of thoracic malignancies. International collaboration in our anticipated exchange program will enable initiation of joint protocols to hasten the development of novel personalized therapies for these neoplasms, and we are indebted to everyone who has worked diligently to support our efforts. Thank you for your time.
Thank you, Dr. Schramm, for the great speech. Our next VIP guest is the president of National Yang Ming Chiao Tung University, Dr. Lin Qihong. Dr. Lin, please.
Mrs. Nina Solarz, Dr. David Schramm, and all the guests here in Taiwan. It is my great pleasure to be here. I'm probably not familiar with most of you because I'm a newly inaugurated president of the newly formed National Chiao Tung University. Let me just introduce myself and the new university a little bit. I am the alumni of National Yangmin University or Yangmin Medical Med School. Alumni of this Yangmin team, I was able to have the opportunity to become the first president of the newly merged National Yangmin Chiao-Tung University. You probably have all familiar with the Yangmin as well as the Veteran General Hospital Taipei. So our new partner is actually from Hsinchu area, Chiao-Tung University. National Chiao-Tung University, I think it's a very renowned university, especially in the semiconductor industry. I think it's suffice to state that National Chiao-Tung University forms the cradle of the Taiwan currently very flourished semiconductor industry. And the entire campus is now moving for the health and medical, especially those for future industry development. So this merger also stands for a new opportunity for the university to not only to continue our in-depth research in the biomedical field, but also to bring in different disciplines to take a multi-modality, multidiscipline research in various topics, including the thoracic oncology that we all heard about today. So I think it's my honor to be here, and also the initiator of this international laboratory, Dr. Muhua Yang. Dr. Yang is currently the provost of the Yangmin campus of the National Yangmin Chiao-Tung University. So he's going to be responsible for continuing our efforts with NCI, with Taipei TVGH, and also continue our effort under the President Liang's supervised National Research Institute of Health, National Health Research Institute, NHRI, also next to Hsinchu area. So I think our coverage area is very much enlarged, and the disciplines involved will be widened. This is a great opportunity, and look into the project scope, the full sub-project elaborated by David. I think this is a very good opportunity for us to focus our efforts on this very important diseases. I believe that this event, this interaction, and also I saw the seminar, hope I can attend all the events. This will be the first of the many upcoming collaborated efforts and production in the near future. Thank you all very much.
Thank you, Dr. Lin, for the speech. Now let's welcome the Superintendent of Taipei Veterans General Hospital, Dr. Wayne Xu-Huiheng. Dr. Xu, please.
So good morning or good evening, Mrs. Lina Solarz, Dr. David Schwamp. I'm Superintendent of Taipei Veterans General Hospital. You can say I'm semi-new for these hospitals. I took a position of the superintendent of this hospital back to January 2021. So before that, I worked in the brother hospital of Taipei TVGH. The name is Taidong Veterans General Hospital, which is located in the central part of the Taiwan. And I'm diabetes doctor. I was also adjunct professor of the National Yangmin and Chiao-Tung University for decades. So on behalf of the Taipei Veterans General Hospital, it's my great honor to welcome our distinguished fellows from NCI, NIH, Dr. David Schwamp, and my outstanding colleagues at Taipei Veterans General Hospital, National Yangmin Chiao-Tung University, as well as Mrs. Lina Solarz, an officer from AIT, and also the Miss Regine Chen, Chen Hui-Zhen, that's the Deputy Director General, Department of North American Affairs of the Ministry of Foreign Affairs in Taiwan, to take part of today's joint symposium. As many of you be aware that Taipei VGH has been the leading medical centers in Taiwan for decades. And one of our major missions is to establish international collaborations with famous esteemed institute and scholars. And we have the state-of-the-art medical facilities and outstanding research group across all the medical disciplines that shape the strong basis to establish all the international collaborations. Other Stephen Solarz Joint International Laboratory is located at the ninth floor of our medical science and technology buildings, where we provide generous laboratory spaces, the first-in-class research equivalent facilities, including the first EDT level laboratory, the world-class clinical research center, as well as one of the largest big data centers, all located nearby. So in the past 2 years, Taipei VGH had provided more than 5 million NT dollars for research fund for these joint laboratories. And recently, lung cancers has been the leading causes of death in Taiwan and many parts of the world. And 3 experts from this hospital will deliver speech and sharing their current research progress today. And we do look forward to exchanging opinions and thoughts from Professor Schramp and Dr. Turamate. And I'm sure this online joint symposium will be a fruitful meeting that can inspire and drive the success of this joint international laboratory between US and Taiwan. So thank you for your listening.
Thank you, Dr. Xu, for the remarks. Apart from the distinguished guest who just gave us remarks, there are still several VIP guests who have made significant contributions to establish this NCI, TVGH, and NYCU international collaboration. First of all, we would like to thank Dr. Steve Guo Song, the former president of National Yangming University, who has made great efforts to initiate the TVGH NYCU lung cancer research team that compete with other outstanding teams in Taiwan and facilitate the cooperation between the two parties.
Good morning, Mrs. Nina Solarz, David, and Randy. And good morning, or I should say good evening to Mrs. Solarz, and good mornings to everyone here in Taipei. There's a Chinese saying, 好事多磨, meaning it takes times to have a truly good things to happen. And I believe this is just one of them. It takes so many times, but like the speakers before me, I mean, really, this is the well-considered and well-intended project. Perhaps I think this is the appropriate time that I tell you another success story of Taiwan and NIH. You know, about 6 years ago, when I was still the Director General of Taiwan CDC, I have the opportunity to foster a collaboration between a local vaccine company and a lab in NIAID under Tony Fauci. Originally on the dengue vaccines, because there's a newly developed vaccines in the NIH, NIAIDs, and we need that, okay? Many years ago, actually, there's a big outbreak in Taiwan, in China especially. And so the collaboration continues to phase 2 studies. And then 2 years ago, the COVID-19 strike. So with the basis of the collaborations and the mutual trust, the team moved to another lab under Tony Fauci, the lab for messenger RNA and also for the COVID-19, Dr. Bonnie Graham. And so was intense talk and technical transfer. And now this company, Metagens, actually has succeeded to produce vaccines for many people to use in Taiwan, including President Tsai Ing-wen and the Vice President Lai Ching-te, and also my mom, who is now 90 years old. Okay. And so that is another good thing. And results from the Taiwan's scientists and industry with NIH. So I'm pretty sure, and I'm so confident that I think down the road, this even better collaboration between David and Randy and Yangming Jiao Tong University and Taipei, I think will come to another great achievement. Maybe at that time I can also use the product as well. But anyway, I'd like to join, the gentleman before me to congratulate and say thank you to you, Mrs. Solarz, for your efforts for this wonderful, wonderful project. And I also have to say that thank you for your late husband, Congressman Solarz, for his help to Taiwan. We will all miss him. Thank you very much.
Thank you, Dr. Guo, for the remarks. Secondly, we would like to thank Dr. Zhang Deming, the former Superintendent of Taipei Veterans General Hospital, who strongly support this collaborative project by providing lab space and setting up funds and equipment for research. Currently, the operation of this international laboratory is under the supervision of the Vice Superintendent, Dr. Hou Mingzhi. Last but not the least, we thank the American Institute in Taiwan and the Ministry of Foreign Affairs for helping us signing the Memorandum of Understanding and the Operating Agreement, which implements and deepens the scientific research cooperation. Therefore, we would like to dedicate a memorial plaque of the Stephen J. Solarz International Laboratory to all these VIP guests whose efforts has made this international cooperation. Please, Dr. Guo, President Dr. Liang, President Dr. Lin, Superintendent Dr. Xu, and the Deputy Director Chen Huizhen副司长of the Department of North America, Ministry of Foreign Affairs, come to the podium and have a photo with the memorial plaque. Thank you. Now I would like to invite everyone in the conference room to come to the front for a group photo with our online VIP guest. Please come to the front for a group photo. Thank you. Deputy Director Chen Huizhen, please stay in the podium, as we would like to present our acknowledgement and ceremonial gift to you for the efforts Ministry of Foreign Affairs has made to strengthen and deepen the bilateral collaboration. Thank you, and please be seated. We will start our scientific symposium in seconds. And please allow me to remind you that we will not take questions in between the talks, but will open for questions at the end of the symposium if schedule allows. For those who wish to raise questions, please enter the link attached below the YouTube broadcast and fill out the questionnaire. We will select some of the important questions from the audience later on. Now the first section of the symposium will be chaired by Dr. David Schramm from National Cancer Institute. Let us welcome him on screen. Dr. Schramm, please.
Dr. Sean, please turn on your mic, please.
Can you hear me now?
Yes.
I am very sorry. It gives me great privilege to introduce the scientific portion of this program this evening by introducing our first speaker, Dr. Shiwa Chu. Dr. Chu received his medical doctorate from the National Defense Medical College, and his postdoctoral training was performed at the City of Hope National Beckman Research Institute. Dr. Chu has focused his research direction on investigating the basic regulatory networks in cancer stem-like properties and pluripotent stem-like cells. He went to Dr. Ralph Reisenfeld's lab in the Scripps Research Institute, where he acquired basic research training in cancer and cancer stem cell biology, establishing a strong background in cancer research. Dr. Chu is currently a professor of the Institute of Pharmacology in the National Yang Chiao Tung University and is director of the Department of Medical Research in Taipei Veterans General Hospital. His talk is entitled Targeting Cancer Stem Cell Network and Cancer Stemness Related Non-Coding RNA in Lung Cancer. Dr. Chu?
It's my great honor to be invited and give a talk in these bilateral collaborative meetings. Today's my presentation topic is Targeting Cancer Stem Cell Network and Cancer Stemness Related Non-Coding RNA in lung cancers. For many tumor types, the de novo mutations leading to primary cancer stem cell are varied. Thus, one would expect that the biology of primary cancer stem cells may also be heterogeneous. Properties such as the cancer stem cell frequency, cell surface phenotype, and the drug sensitivity may vary as a function of the specific mutation in which the primary events occurred. Notably, neoplastic progression may occur either as a consequence of intrinsic tumor pathogenesis and challenging with chemotherapy. Cell renewal and pluripotency are the central features in the definition of embryonic stem cell, in which OFO-SUC2 plays a key role in the maintenance of this stem cell property. Recent publications shows embryonic stem cell-like genes was highly detected in the poorly differentiated aggressive tumors, and with parallel the worsened the clinical outcome. On the contrary, the stem cell markers, including OFO, SUC2, CAFO, CMYC, and LIN28, not only maintains the stem cell function, but also can reprogram the adult cell, reprogramming into the pluripotent status, so-called induced pluripotent stem cell. So, the Stemness gene signature in cancers in stem cell maybe have a different, play the important role, but a different the functions. So, based on these findings, we would like to explore whether a cancer stem cell existed in the clinical samples and whether the cancer stem cell property and the mechanism involved in cancers. Our approach for isolating and expansion of the cancer stem cell is according by the sphere formation by serum-free selection medium. Here we show the data can show that the tumor sphere is highly enriched by the serum-free medium, and also the cancer stem cell marker, including CD133, are highly evaluated. More importantly, we highly detect the embryonic stem cell marker, including the OCD4 NANOC in this tumor sphere enriched by the serum-free medium. And most importantly, we find that these findings is similar happened in the clinical samples, including OFO NANOC co-relocalized with the CD133 positive surface marker. Most importantly, the highly co-expressed the OFO NANOC and the CD133 cancer stem markers, the group the patients present the most worsened clinical outcome. Furthermore, we further to test OFO the function in the CD133 lung cancer stem cell, and found that when knocked down the OCT4 expression in CD133 cells, we can find not only the OFO expression is greatly decreased, and you can see the cancer stem cell phenotype, the tumor sphere also diminished, and importantly, the CD133 cancer stem cell markers infiltration level and the total subpopulation also significant decrease. Therefore, we would like to know whether the OFO and the nano co-express can help the cancer stem cell property, and we using the lung cancer, lung carcinoma cell line, and the co-express OFO and the nano. The overexpression, the data shown can enhance the cancer stem property, and also in the compared to the control cell, the CD133 positive population is highly elevated. It's according by the OFO and the nano co-express. Importantly, our using this OFO nano co-expression cell line nano transplant into the immune-compromised mice, and the data shows this kind of co-expressed OFO nano cell can promote in vivo tumor regenic and metastatic ability. And also you can see the IHC finding shown the highly mitosis and also shown some of the differentiation reprogrammed the phenotype in this xenograph. Finally, we knock down of OFO and nano expression in these cancer stem cell models, and the result shows this kind of knockdown can effectively block the metastatic ability of OFO nano expressed lung adenocarcinoma xenograft in animal models. So we make a conclusion: the embryonic stem cell markers, including OFO nano, can induce the cancer stem-like property and enhance proliferation, including ENT property, and contributing to the tumor genesis and metastasis in lung adenocarcinoma. We therefore propose that the OFO nano and the ENT transcription factors will be the potential markers of prognosis and a novel target of therapy for lung adenocarcinoma treatment. Circular RNA are non-coding RNA derived from the base splicings of the pre-messenger RNA to give a closed as a liquid biopsy biomarkers for lung cancer progressions. Furthermore, based on our NGS and the bioinformatic analysis, our data further shows that the OFO is one of the upstream candidates and can modulate circular RNA expression in cell line. We further detect whether this scenario will happen in the real lung cancer status. So we take the clinical samples and using the immunohistochemistry combined with fluorescent in situ hybridization and the digital droplet digital PCR, and our data shown here. Here in this slide, we further confirmed C190 upregulation in lung cancer's clinical tissue samples using the RNA FISH and the ddPCR. Importantly, our observation was and the findings are including the tumor samples showing higher C190 expression as compared to the adjacent normal lung tissue samples. So this data support C190 is also overexpressed in lung cancer carcinoma and the clinical samples, especially compared to the normal lung tissues. To further explore the potential roles of a C190 in the lung cancer, especially in the cancer growth or even in the proliferation, we further using the C190 overexpression models in vitro and then the overexpressed stable clone transplanted into the immunocompromised mice to establish a xenograft model. And our data shows C190 overexpression could increase the tumor growth and the proliferation in this kind of xenograft model. And most importantly, our FISH models can further detect by the fluorescent in situ hybridization was used to confirm the C190 expression in the xenograft tissues. Importantly, the correlate similar consistent data, the KICC7 proliferative markers are also shown in increased expression in the C190 overexpressed xenograft tumors as compared to its control. Taken together, all of these results, we prove that the C190 in vivo upregulation can support higher tumor growth. CRISPR-13A is an endonuclease enzyme guided by the bond of CRISPR RNA and the circular RNA to target RNA transcript for editing degradation. Recently, is have successfully using this CRISPR-13 system to either detect RNA or edit RNA. Based on this previous suggestion and the comments, we further in this study, we used the CRISPR-13A as a target, the C190 for degradation. So this is our whole study experiment design. We using the CRISPR-13A as a model to mediate C190 targeting and in vitro, and we established the model, and then using the models in transfer to in vivo nanograft. All data shown prove that these CRISPR-13A C119 targeting cells or lasers the tumor size and the smaller one, and also the tumor volume significant decrease. Importantly, we further use the ddPCR to detect the C190 expression, and the result to confirm that the CRISPR-13A C190 targeting cells express lower C190 level. Therefore, our results suggest targeting C190 can be an effective strategy, especially potential for the retractable lung cancer treatment. Most importantly, we are using FISH to detect the C190 express, and can confirm and also support our CRISPR-13A mediate C190 effect, and also can compare to the Ki-67 when using the CRISPR-13A mediate C190 targeting. The Ki-77 expression also decrease. So we make a conclusion here. We identify the circular RNA 190, a non-coding RNA with a pathogenic function, as a negative prognostic biomarker for lung cancer upregulated in lung and non-cancer cell line. And using the transient and stable overexpression of C190 in lung cancer cells, resulting in increased pro-oncogenic phenotype, and also increase the tumor growth in mouse xenograft. Importantly, we successfully applied the novel CRISPR-13A system to knock down the C190 expression, and demonstrate that the knockdown significantly curbed to its tumor genetic property. We thank our colleagues from the National Yang Jiao Tong University, and also we teammates in the TVGH, Taipei Veteran Children's Hospital, the chest medicine, Director Chen, and the surgical department chief, Xu, Dr. Xu, and my lab, the postdoc, and the technician and students work together to contribute to this research work. And I thank you everyone to take attention to. I would like to take the question for all. Thank you for your attention again.
Thank you, Dr. Xu, for your very enlightening speech. Our next speaker is Dr. Yumin Chen. He is currently a clinical professor at the National Yang Jiao Tong University and Taipei Medical University. Professor Chen obtained his PhD from the Institute of Clinical Medicine at National Yang Tao University. He's been a visiting fellow at the National Cancer Institute in Bethesda, USA. Professor Chen is a renowned pulmonologist and is head of the Department of Chest Medicine at Taipei Veterans General Hospital. He has dedicated his career to the diagnosis and treatment of lung cancer. His area of research includes targeted therapy, chemotherapy, immunotherapy, and tumor immunology. Professor Chen also has been the principal investigator of many global cancer trials. His talk is entitled Personalized Precision Therapy in Non-Small Cell Lung Cancer. Dr. Chen?
Dear Chairperson and colleague, it is my pleasure to have a brief talk in this meeting. I'm Chen Yuming from Department of Chinese Medicine, Taipei Veterinary General Hospital. The title of my talk is Personalized Precision Therapy in Non-Small Cell Lung Cancer. From 1986 to 2017, the overall age standardized incidence of lung cancer in Taiwan is increased from 22 to 44 in men and from 9 to 32 cases in women per 100,000 populations, respectively. The age standardized mortality rate also increased from 28 to 32 deaths in men and from 14 to 16 deaths in women, respectively. This figure will demonstrate that lung cancer precision therapy improves stage 4 non-small cell lung cancer patients' survival with molecular target therapy and immunotherapy, and how important the precision medicine is. With the advancement of precision oncology, including target therapy and immunotherapy, current practice in advanced non-small cell lung cancer include checking tumor driver mutation status and tumor PD-L1 expression, and then treatment was given according to these findings. While there are different proportions of tumor driver oncology between Asian and the US or Europe, such as EGFR mutation status are more frequently found in East Asia, while KRAS mutation are most frequently found in Western countries. For example, standard of care for EGFR mutant non-small cell lung cancer in first-line treatment include osimertinib, dacomitinib, afatinib, orotinib plus bevacizumab, or orotinib plus ramucirumab. And there are a broad range of resistance mechanisms in EGFR mutant non-small cell lung cancer following the failure of EGFR TKI therapy. For example, secondary T790M mutation accounts for 60% of resistance after first-line orotinib, gefitinib, or afatinib treatment. We published an article last year that clearly showed the importance of the biopsy for tumor molecular analysis, such as in this study of T790M second-line mutation. Patients who received first-time biopsy had a 53% positive for T790M mutation, while repeated biopsy in those patients whose first-time biopsy was negative for T790M mutation showed 37% positive rate for T790M mutations, and the survival curve clearly showed patients who had T790M mutations and received third generation EGFR TKI osimertinib had the best survival, while those with T790M mutation without osimertinib treatment, or those patients without T790M mutation had a poor survival. Liquid biopsy molecular examination is another important tool for lung cancer treatments, such as this patient who was suffered from repeated disease progression after effective first-generation EGFR TKI gefitinib treatment, and her plasma cell-free DNA showed T790M mutation. On September 2016, she then received osimertinib and the chest X-ray 3 weeks after starting osimertinib showed markedly treatment response and had a progression-free survival of 19 months. This is another example of using liquid biopsy molecular analysis of CSF for choosing appropriate patients who receive third generation TKI in patients with leptomeningeal carcinomatosis, and the results were just published this year. Now we are performing NGS analysis of CSF both before and after third generation TKI treatment for detecting resistance mechanism of third generation TKI in CNS metastasis, including meningeal carcinomatosis. We also published a series of review articles regarding immune checkpoint inhibitors and the importance of combination immunotherapy since 2017. We recently reported comparison of the outcome between immunotherapy alone or immunotherapy in combination with chemotherapy in EGFR mutant non-small cell lung cancer, and we found that T790M mutation status might be a useful predictor for poor response to treatment comprising immunotherapy alone or immunotherapy combined chemotherapy. Again, demonstrated that appropriate molecular marker is important for better treatment choice, both in target therapy and immunotherapy. Normal molecular target therapy for non-small cell lung cancer evolved rapidly, and the FDA already approved 7 novel agents for specific tumor driver mutations. Take MET as an example. We also actively involved in clinical trials on MET exon 14 skipping mutations and MET amplification, and the results are just published in New England Journal of Medicine and Lancet Respiratory Medicine last year. Recently, we have a 4-year project on Ministry of Science and Technology of Taiwan sponsored proposal establishing integrated medical big data for the translational research of non-small cell lung cancer and the head and the neck squamous cell carcinoma. And this project is approved since June of this year. Specific aims include to collect clinical, radiological, pathological, and genomic data of non-small cell lung cancer and the head and neck squamous cell carcinoma patients to elucidate the unique genomic landscape to establish an integrated platform consisting of clinical, radiological, pathological, and the genomic data, and to explore biomarkers and constructing risk prediction models for this patient. Clinical groups enroll patients according to inclusion and exclusion criteria. The Department of Radiology is responsible for CT image data collection, and the Department of Pathology is responsible for digital pathology and scanning and cancer genomic study. The Department of Informatics will perform data integration. Finally, through the presentation learning and the statistical analysis of this heterogeneous data, we try to transform them into predictive markers or risk prediction models, and the prediction treatment for these patients will be given accordingly. That's all my brief talk. Thanks for your attention.
Thanks for the informative speeches. Our next talk will be chaired by Dr. Yang Muhua. Dr. Yang, please.
Good evening and good morning, dear colleagues. It's my great honor and pleasure to have the chance to chair the following section. The following section, the first speaker is Professor David Trump. Professor David Trump graduated from the University of Connecticut School of Medicine and completed his general surgery residency at the University of Chicago, as well as completed research fellowship in Memorial Sloan Kettering Cancer Center. And then he received the thoracic surgery residency at the University of Michigan. Then Professor Trump was appointed to MD Anderson Cancer Center, be the faculty member of cardiothoracic surgery. In 1997, he was appointed and moved to NCI to lead the team of thoracic oncology surgery branch of NCI till now. And he is a renowned thoracic surgical oncologist, and is also an outstanding expert in the field of genetic regulation and the cancer stemness of the thoracic malignancy. Welcome, Professor David Trump.
Thank you for that introduction, Muhua. What I would like to do today is to briefly discuss our attempts to utilize induced pluripotent stem cells to model and target thoracic malignancies. I have no conflicts of interest. In addition to our clinical responsibilities in the thoracic surgery branch at the National Cancer Institute, a major emphasis of our laboratory work is focused on epigenetic effects of cigarette smoke and other environmental carcinogens, with particular emphasis on those epigenetic alterations that induce pluripotency or stemness in cancer cells. We have focused primarily on DNA methylation, polycomb repressive complexes, and non-coding RNAs as potential therapeutic targets in lung and esophageal cancers and malignant pleural mesotheliomas. The strategy that we have used for developing novel epigenetic therapies for thoracic malignancies is summarized in this slide. We have sought to fully utilize the tumor tissues that we have resected from our patients, and we have developed reliable means of isolating stem-like cells from these surgical specimens, as shown here by spheroids grown out of these tumor specimens, either on non-adherent conditions or on Matrigel. We can also take normal cells from the lung, the esophagus, or the pleura and reprogram these cells to make induced pluripotent stem cells, or iPSCs, which we can further manipulate through gene editing techniques, or we can expose to cigarette smoke and other environmental carcinogens. This allows us to perform comprehensive genome-wide analysis in an integrated manner to look at epigenetic landscapes, multi-step environmental carcinogenesis, and plasticity of the various cells that we have generated. The ultimate goal is to develop novel pharmacologic and immunologic regimens that we can test in clinical trials to develop more effective therapies for cancer. In our previously published work, we have demonstrated that cigarette smoke mediates time-dependent changes in core histone proteins, as well as DNA methylation, mRNA, and microRNA signatures that coincide with progression to malignancy of respiratory epithelial cells. We have also demonstrated that cigarette smoke markedly increases the malignant phenotype of lung cancer cells in vitro and in vivo. Our current smoke exposure experiments have utilized fully differentiated cells for the in vitro analysis, in which the smoke exposure is believed to enable a de-differentiation back to an abnormal epigenetic state and perhaps allow outgrowth of stem cells or progenitor cells from these cultured cell lines. As we've thought about this more recently, we've wondered whether or not it might be also appropriate to try to use stem cells or progenitor cells for the smoke exposure experiments to facilitate outgrowth of these pluripotent stem cell populations. However, in our experience, it's been very difficult to isolate and propagate these stem cell progenitors, so we wondered whether or not induced pluripotent stem cells could help elucidate epigenomic events associated with carcinogenesis and identify novel therapeutic targets in thoracic malignancies. In order to examine this issue, we reprogrammed normal human small airway epithelial cells to pluripotency by transduction of Yamanaka factors. In this left-hand panel here are the parental small airway epithelial cells, and the remaining photographs show the sequential changes during the reprogramming process until there is outgrowth of induced pluripotent stem cells. These iPSCs exhibit characteristic patterns of stem cell gene and stem cell antigen expression and form typical teratomas when inoculated into immunodeficient mice. Comprehensive analysis demonstrated that there were significant changes in DNA methylation signatures as the cells reprogrammed from small airway epithelial cells to lung-induced pluripotent stem cells, as shown here in these graphs, and that there were 15,000 genes or more that were differentially expressed as a result of the reprogramming from normal small airway epithelial cells to pluripotency. A major question pertaining to all of this work is how closely the lung-induced pluripotent stem cells reflect gene expression profiles in human lung cancer. And in order to examine this issue, we evaluated gene expression profiles in a panel of small cell lung cancer and non-small cell lung cancer lines relative to induced pluripotent stem cells from small airway epithelial cells. And then all of these cells here were normalized back to parental small airway epithelial transcriptomes. On the right side is a Venn diagram which depicts the number of genes that were unique or shared among the three different cell types. What we observed was a substantial number of genes, nearly 44%, are uniquely expressed in the lung-induced pluripotent stem cells. 1/3 of the genes that were differentially regulated in the lung-induced pluripotent stem cells were modulated in a histology-specific manner. And interestingly, although the lung-induced pluripotent stem cells were derived from small airway epithelial cells, the signatures of the lung-induced pluripotent stem cells overlapped much more with neuroendocrine small cell lung cancers compared to non-small cell lung cancers, and this was a 19-fold difference. One of the genes that we identified as being differentially expressed during reprogramming of small airway epithelial cells to pluripotency is ASXL3, which encodes a poorly characterized protein that is believed to be involved in regulation of chromatin structure. In the left-hand panel here are results of quantitative RT-PCR as well as immunoblot experiments demonstrating upregulation of ASXL3 at the mRNA and protein levels in induced pluripotent stem cells with no evidence of expression of ASXL3 in normal small airway epithelial cells. When we further examined ASXL3 expression in a broad panel of cultured normal respiratory epithelial cells as well as non-small cell and small cell lung cancer lines, we observed marked upregulation of ASXL3 in small cell lung cancer lines. We validated these findings using immunohistochemistry techniques and observed a significantly higher expression of ASXL3 in primary small cell lung cancer specimens relative to non-small cell lung cancers with no detectable ASXL3 expression in normal lung tissues. CGH array analysis suggested that the magnitude of ASXL3 upregulation in small cell lung cancers was associated in a significant manner with gene copy number, suggesting that this region of chromosome 18 that encodes ASXL3, a region not previously known to be associated with lung cancers, is actually amplified. When we knocked down ASXL3 in the induced pluripotent stem cells, we saw a significant reduction in the frequency and size of subsequent teratomas. And when we knocked down ASXL3 expression in small cell lung cancers, we saw significantly reduced in vitro growth as well as growth of small cell lung cancer xenografts in athymic nude mice. Collectively, this constituted the first reported observation that ASXL3 is upregulated in stem cells, and the first demonstration that ASXL3 overexpression contributes to human cancers. Functional analysis of ASXL3 is underway, and numerous other genes identified during this transcriptome analysis are the focus of ongoing experiments. In additional experiments, we have sought to examine whether or not we can transform lung-induced pluripotent stem cells to malignancy to better model cancer stem cells. To examine this issue, we use flow cytometric techniques to isolate the pluripotent side population from cultured small cell lung cancer lines, and transcriptome analysis suggested that these pluripotent stem cells were intermediary between normal stem cells and the small cell lung cancers. We have further sought to push normal lung-induced pluripotent stem cells toward malignancy by exposing them to cigarette smoke, with the intention of establishing a continuum from normal induced pluripotent stem cells through cancer stem cells to a heterogeneous small cell lung cancer population. Collectively, these experiments may identify those genes or pathways that are essential for maintaining stemness in lung cancer cells that can be novel therapeutic targets. Encouraged by the results of our lung-induced pluripotent stem cell experiments, we have recently generated esophageal-induced pluripotent stem cells in an attempt to model esophageal carcinomas. In this upper left-hand panel are photomicrographs demonstrating normal esophageal epithelial cells before reprogramming and on day 7 and day 21 of the reprogramming process. This panel depicts the results of RT-PCR analysis demonstrating expression of the transduced Yamanaka factors on day 7, with silencing of these transgenes subsequently as the reprogramming progresses. The induced pluripotent stem cells exhibit characteristic phenotypes in terms of stem cell antigen and stem cell gene expression, as shown by these confocal experiments, and the induced pluripotent stem cells form teratomas when inoculated into immunodeficient mice. In additional experiments, we have developed methods for reproducible isolation of stem-like cells from primary thoracic malignancies. In this upper left-hand panel are representative photomicrographs of stem-like cells that we have isolated from colorectal carcinoma metastases to the lung, as well as from lung cancers, malignant pleural mesotheliomas, and esophageal adenocarcinomas. These stem-like clones exhibit characteristic alkaline phosphatase staining, as well as prototypic expression of stem-like markers consistent with a stem-like phenotype. These cells also exhibit marked resistance to chemotherapeutic agents such as cisplatin, as demonstrated in this dose-response experiment. Because we are now able to reproducibly isolate cancer stem-like cells from primary thoracic malignancies, we can compare transcriptomes of the lung and esophageal-induced pluripotent stem cells with the transcriptomes of cancer stem-like cells isolated from primary lung and esophageal cancer cells. We can identify genes that are commonly regulated, but more importantly, we can identify genes that are only expressed in the cancer stem cells, as depicted by the asterisks in this Venn diagram. We can then use DNA methylation array techniques to see whether or not these unique signatures of the cancer stem-like cells correspond with specific DNA methylation signatures in these cells. And furthermore, we can perform comprehensive genome-wide analysis using ATAC-seq or DNase hypersensitivity sequencing with DNA footprint analysis to identify transcription factor binding sites that are unique only for the cancer stem cells. By combining all three techniques, we can perform comprehensive genome-wide functional analysis of potential targets. And this approach has already demonstrated to us in unpublished experiments that chromatin accessibility is the real issue. We can also use the lung and esophageal-induced pluripotent stem cells and corresponding cancer stem cells isolated from primary lung and esophageal cancers for high-throughput drug screening as well as autologous typing and antigen discovery experiments, hoping to identify pharmacologic or immunologic regimens that kill the cancer stem cells while allowing the induced pluripotent stem cells to remain healthy and strong. In summary, induced pluripotent stem cells can be used as models to identify novel mediators of stemness that are potential therapeutic targets in thoracic malignancies. Although I did not have a chance to describe these experiments fully, lung and esophageal induced pluripotent stem cells exhibit common as well as distinct transcriptome signatures that may reflect tissue of origin and/or extent of reprogramming. Combined analysis of the lung and esophageal induced pluripotent stem cells and corresponding cancer stem cells from surgical specimens provides unique opportunities to develop highly specific and theoretically less toxic pharmacologic and immunologic regimens for thoracic malignancies. I would like to acknowledge additional outstanding members of the thoracic surgery branch who have directly contributed to our translational research efforts, including an exceptional team of scientists who perform the experiments that I have described today. I'd also like to thank our courageous patients without whom none of this would have been possible. Thank you for your time.
Thank you, Dr. Schramm, for the wonderful speech. Our next talk will be chaired by Dr. Qiu Shihua. Dr. Qiu, please.
Good evening, everyone. It's my great honor to introduce the next speakers. Next speaker is Dr. Tuana Tolonet. She's I brief introduction about her the bio. Dr. Tuana Tolonet is a physician graduated from the Spanish University in the Istanbul and has served in the Department of Public Health of Turkey. She commenced the Solarz fellowship program in July 2020, and her work is focused on the lung cancer stem cell research in the thoracic surgery branch at National Cancer Institute. Today, she will bring the topic is modeling the Stemness and the metastatic potentials of lung cancer cells in vitro and in vivo. So now welcome Dr. Tuana Tolonet. Thank you. That's welcome.
Modeling stemness and metastatic potential of lung cancer cells in vitro and in vivo. Cancer stem cells are believed to be the precursors of many tumorigenic phenomena, in which metastasis is the leading concept. Some theories accept that after metastatic cascade begin with the primary tumor and its environment, cancer stem cells, which are the precursor circulatory tumor cells, detach from the primary tumor, intravasate into circulatory systems, evade the immune attack, extravasate in distant capillary beds, and finally invade and proliferate in distant organs. This mechanistic theory, however, is not enough to explain by which exact epigenetic modifications happen in and around the tumor tissue. Therefore, many researches have been done to isolate cancer stem cells from primary tumors. We additionally created a syngeneic immunocompetent mice model with C57BL6 mice using murine lung cancer cell line to study different pathways and targetable mechanisms which can be attributable to metastatic cascade actions and can be novel treatment options for metastatic lung cancer. To perform this study, we isolated 24 stem-like clones from parental Lewis lung cancer cell line. We used low attachment culturing conditions for maintenance. However, experiments have been done under two-dimensional conditions. Selected clones were tagged with luciferase, and bioluminescence imaging has used to screen the tumor formation and metastasis. To have more purified clones which have restricted organ-specific metastasis, we isolated enriched clones. To do that, we dissected tail vein-injected tumor-bearing mice, chop and mince the tumor tissue, wait for coming cells to attach and grow in the media, select the luciferase tag cells and kill the other normal cells with some specific antibiotics, then use them for further injections and culturing. After establishing different clones, we assess their stemness features. Here, on the left upper side, you can see the quantification of spheroid formation assay and different numbers of spheroids in each clones. Right next to it, you see the different expression patterns of well-established cancer stem cell surface marker CD133 between our clones and the increase in fold changes compared with the parental cell line. Additionally, here is the qRT-PCR data showing highly expressed cancer testis antigen in our clones. As one of the further stemness features, clone 8 and clone 9 cells show increased resistance to different concentrations of cisplatin drug treatment and discrete growth patterns under in vitro conditions. Serial dilutional injections to NSG mice also showed even 500 clone 8 cells can create a tumor in vivo. However, the minimum amount of cells needed is 10,000 for parental cell line. When we move to the in vivo studies, first we use immunodeficient NSG mice injected subcutaneously with clone 8, clone 9, and parental cell lines. After we euthanized all mice at day 18, we saw clone 8 and parental cell line metastasized to the lung. However, clone 9 cells metastasized to the lung and liver at the same time. As clone 8 and parental cell line showed similar patterns with immunodeficient mice, we then injected them to immunocompetent mice with the same concentrations. These results also showed both cell lines metastasized only to the lungs. However, their in vivo growth patterns were different from each other. While parental cell line tumors keep growing with increased rate, clone 8 cells having a plateau after 22nd day, which is in contrast with the accelerating in vitro growing conditions. This indicates these clones are also promising for future immunology and tumor microenvironment studies. Afterwards, here you see 3 different cell lines injected into B6 mice via tail vein. At day 21, we see parental cell line injected mice has the lowest signal in lungs, while clone 9 cells injected mice show broad multiple organ metastasis. Detailed analysis of those injections showed parental cell line showed lung and liver metastasis with the first round of injection. When we take the lung cells and enrich them with the aforementioned methods, the second round of injection showed lung, thoracic wall, and brain metastasis. And the third round of injection showed thoracic wall tumor cells metastasized to the lung and thoracic wall. Same with the liver cells. After enrichment, they show metastasis in the lung, vertebrate, and liver. Further enrichment of liver cells showed metastasis in the lung and liver. While parental cell line has this diverse capacity of metastasis, after 3 rounds of injections, C8 cells only showed metastasis in the lung. On the other hand, with the same fixed cell concentrations and injection methods, clone 9 cells metastasized to the lung, bone, liver, skeletal muscle, and pancreas. After enrichment, bone tumor cells show metastasis to the lung, liver, skeletal muscle, bone, pancreas, and adrenal gland. Further survival analysis of those injections also showed mortality rates of clone 9 cells are higher than clone 8 and parental cell line. Additional preliminary results of enriched cells subcutaneous injections to B6 mice also showed consistency with our results. For example, when we use second rounds liver-enriched parental cells and inject into B6 mice, we see the spontaneous metastasis to the lung and liver at the same time. After getting these consistent results with statistically significant sample size experiments, we analyzed our cells with RNA sequence and DNA methylation array. When we look at the results of RNA sequence principal component analysis, shows that our clone 8, clone 9, parental cell line display distinct gene expression patterns consistent with the following DNA methylation assay. Here on the left side, you see the schematic display of up and down regulated genes quantifications for each cell line. We see thousands of genes overlap between all 3 cell lines, which is not very unexpected. However, when we look at the specific pathways and genes of each clone, which are the areas painted with different colors and corresponded in the right-sided heat map here, we can see the different and distinct patterns for each clone, which is also supported by DNA metalomes. These heat maps also show the enriched cells, RNA sequence, and DNA methylation assays results. For example, the cells which metastasize to thoracic wall enriched from thoracic wall tumor do show clearly different transcriptomic pattern from liver-enriched liver tumor cells, even though both are from parental cell line. Also, DNA methylation assay shows C8 lung tumor enriched from lung tumor cells have obviously different methylome pattern than C9 bone to muscle cells. In conclusion, our established clones from Lewis lung cancer cell line showed distinct phenotypic profiles in vitro and in vivo, supported by transcriptomic and methylome analysis. Also, they have a potential for novel immunotherapy and vaccine trials. They also provide unique opportunities to examine mechanisms governing metastatic potential of lung cancer cells and evaluate potential efficacy of novel pharmacologic and immunologic regimens targeting cancer stem cells for lung cancer therapy. Thank you for listening.
Thank you, Dr. Tolone, for sharing an informative speech. The next talk will be chaired by Dr. David Schramp again. Dr. Schramp, please.
Thank you. It is my honor to introduce our next and last speaker, Dr. Muhua Yang. Dr. Muhua Yang got his MD and PhD at the National Yang Chiao Tung University and received clinical hematology and oncology training at Taipei Veterans General Hospital. He now serves as the Senior Vice President and Provost of National Yang Chiao Tung University and is also the Director of the Division of Medical Oncology of Taipei Veterans General Hospital. His major interest is the treatment and mechanisms of metastasis of upper airway digestive tract cancers. His talk is entitled Immunomicroenvironments of Upper Aerodigestive Tract Cancer in Taiwan. Muhua, please.
Dear colleagues, today is my great pleasure and honor to have the chance to present my result about the immunomicroenvironment of the upper aerodigestive tract cancer in Taiwan. The upper aerodigestive tract cancer include the cancer originated from oral cavity, pharynx, larynx, and the esophagus. And these cancers share common etiological factors, including smoking, alcohol, and the betel nut consumption. And these factors are similar to the etiology contributed to squamous cell carcinoma of lung. Therefore, we are especially interested in the immunomicroenvironment and the genomic profile of the upper aerodigestive tract cancer in Taiwan versus the Western countries. We are especially interested in the esophagus cancer and the pharynx cancer. So to understand the genomic alterations of the upper aerodigestive tract cancers in Taiwan, we use whole exome sequencing to analyze the genomic alterations in 35 samples of the upper aerodigestive tract cancers in Taiwan. And the TP53 and FAT1, caspase A, CDKN2A, Notch1 are the most altered genes in the upper aerodigestive tract cancers. And here is the mutational map of the TP53, and you can see there's no special hot spot for mutation. And the somatic mutation rate of TP53 is relatively high, nearly 75% of all samples. And here's the CDKN2A. The mutation rate is also high, about 30% of patients harbor the CDKN2A mutations in their tumors. And FAT1 is an intracellular junctional gene, and recently attracted the attention of the biologists that FAT1 is an important protein that mediates the so-called hybrid epithelial mesenchymal status, and a significant proportion of patients also have the FAT1 mutation. In addition to the major mutation gene, we also noticed that the major characteristics of the upper aerodigestive tract squamous cell carcinoma is local invasion rather than distant metastasis. As you can see, in the PET scan show the significant local invasion of the upper aerodigestive tract cancer without systemic metastasis compared to the common metastatic tumor in the breast cancer shown in the right panel. And we previously have found a major mechanism that regulate the collective migration of the upper aerodigestive tract squamous cell carcinoma by upregulating the intercellular junctional protein Claudin 11 to drive the collective cell migration and to form the circulating tumor cluster, and both of them contribute to the systemic dissemination of the tumors. And we further characterize the microenvironment of the primary and the metastatic tumors, and we found the distinct phenotypes of the primary versus metastatic tumor-associated macrophages were noted. In the primary tumor, the co-expression of the M1 marker and the M2 marker was noted. In the metastatic tumor in countries, most of the tumor-associated macrophage are M2-like cells, indicating that the metastatic macrophages are mostly M2-like phenotype. And then we use animal experiment to prove the importance of metastatic macrophage in the disease dissemination. We deplete the metastatic macrophage, and then we found the systemic dissemination of tumor significantly reduced. And then we further found that the increase of the secretion of cytokine interleukin-35 was noted in the metastatic macrophage, indicating that the metastatic macrophage may secrete interleukin-35 to facilitate the metastatic colonization. And we use the animal experiment by neutralizing the IL-35 and the anti-macrophage antibody. And we found neutralization of interleukin-35 represses metastatic colonization of the tumor. And if we give both anti-IL-35 together with anti-CSF1 receptor antibody and macrophage suppressing antibody, and the effect was increased. And furthermore, we want to understand the epigenetic profile of the primary versus metastatic macrophage. And the results show the DNA methylome between the macrophages from the primary versus metastatic tumor are distinct, indicating that the macrophage residing in the metastatic tumor should have a different gene expression signature to regulate disease progression. And we further use the clinical samples from the upper aerodigestive tract cancer to decipher the different gene expression profile. And we cut the tumor as the invasive front versus inner tumor core. And we also collect the paired metastatic tumor, and then we performed RNA sequencing to identify the gene expression signature in the different part of the tumor. And then we calculate the EMT score, the epithelial mesenchymal score, and we found a gradually increased of the EMT score from the inner tumor to the outer tumor to the metastatic tumor, alongside with the metastatic journey, indicating that EMT is a major mechanism involved in the metastatic journey of the upper aerodigestive tract cancers, and the reversal phenotype, i.e., the mesenchymal to epithelial transition that has been noted in the metastatic tumor was not obviously noted in the metastatic tumor in our samples. And then we use the CyberSort bioinformatics analysis to profile the tumor-infiltrated lymphocyte in the RNA-seq upper aerodigestive tract cancer samples. And we found the M2 macrophage signature is dominant in the metastatic tumors, which is consistent with our previous study, just I have shown in the first several slides of my presentations. And then we use another technique, the spatial transcriptomic analysis, to investigate the transcriptomic changes of the different part of the tumors of the upper aerodigestive tract cancer samples. So we select 2 patients and harvest the fresh samples and cut into the inner tumor and outer tumors as we have done in the previous slide. And we use the spatial transcriptomic analysis technique. And this is the case 1, and the panel A is the most outer part, following by panel B and C and the panel D. And then we perform the single cell transcriptomic analysis and use UMAP to cluster the cells. And then we can see there are several outer tumor clusters, and then there are also some stroma that is the infiltrated immune cell clusters. And we can use this tumor cluster to find the corresponding histology in the lower panel to see which is the invasive tumor clusters and which is the inner side tumor clusters and which is the infiltrated immune cell clusters. And for these patients, we use the trajectory analysis to demonstrate the evolution of the inner tumors alongside to the invasive front of the tumor, indicating a continuous transcriptomic evolution from the inner tumor to the outer tumor. And then we also analyze the EMT signature, and we can see also a significant change of the EMT signature. And then this is the second patient. We also cut tumor samples as the inner side and the outer part. And then we also use UMAP to cluster the sequencing result. And then there are also several tumor clusters, and we also have several immune cell clusters. Compared to the first patient, the second patient, the samples is abundant of the immune cell infiltration. Therefore, we can further analyze the immune cell infiltration in these samples. And therefore, we especially pay attention to the tumor inflammation signature, TIS. And we can see the different tumor clusters has different tumor inflammation signature. And the cluster 1 relatively higher compared to the cluster 2 and the cluster 3. So you can see the cluster 1 is the outer margin of the tumor, has a relatively higher tumor inflammation score, indicating a relative high inflammation signature in the invasive front tumor. We further analyze the surrounding immune cell by this spatial transcriptomic analysis. And we can see the surrounding immune cell in cluster 1 is relatively abundant comparing to the cluster 2, that is representative of the inner side of tumor. And the cluster 1 and heavy infiltration of T cells noted. In the cluster 2, you can see the infiltration of neutrophil and the macrophages. So I would like to summarize of my presentation today. The first is the upper aerodigestive tract cancer is a major type of aggressive cancer in Taiwan with unique genomic, clinical, and transcriptomic features. This cancer tend to invade locally, has some featured mutation, and the transcriptomic feature is also unique. And the second is we found the metastatic tumor-associated macrophages are distinct from the macrophages from the primary tumor, and they secrete interleukin-35 to facilitate the colonization of metastatic tumors. The third is that we use spatial transcriptomic techniques, and we found distinct gene expression profiles between the invasive front and the inner part of the tumor, and furthermore also denoted in the metastatic tumor alongside with the journey of the metastasis. And the tumor inflammation signature and the infiltrated immune cell are significant, closely related to the EMT phenotype of the upper aerodigestive tract cancer, and the cancer located in the invasive front have a higher inflammation signature and the heavy infiltration of immune cell, indicating a significant tumor stroma indication, and the inflammatory effect was noted in the invasive front of the upper aerodigestive tract cancer. I would like to thank to my lab member and the previous member and my collaborator from the National Yang Jiao Tong University and the Taipei Veteran General Hospital and the Makai Memorial Hospital for providing the precious technique support and the clinical samples and the precious clinical information and guidance. And thank you for your attention. And welcome to visit our new merged university, the National Yang Jiao Tong University. The left photo is from the Taipei district, Taipei campus of the NYCU. The right is the photo from the Hsinchu campus of the NYCU. Thank you for your attention.
Thanks for all the inspiring presentation. Now we have only a little bit of time for one question for both sides. Dr. Yang will be the chair.
Here we receive a question from the audience. And the question is about the future perspective and the collaboration of the joint lab. So I would like to invite Professor Schramm and Professor Chiu and the Vice President Wei to represent the three institutes to respond to the audience. So, Professor Schramm.
Can someone tell me the question, please?
Question is about the future perspective and the collaboration of this new joint lab, which may be the first one of the Taiwan and the United States joint lab.
So I think that that Gangi Liang really summarized it very well. This was a tremendous amount of work by a very large number of people, during which we got to know each other, we established friendships, we established trust, we share a common vision in terms of the goals and perspectives of the work that we hope to achieve. I think that this is a really unique opportunity based on the complementarity and the potential synergy of the various groups and the strengths of the three institutions. I think that this will take a lot of hard work, there is no question, but we have very unique opportunities, I think, to make potential breakthroughs in cancer therapy.
I think I would like to invite Professor Chiu to say something about the International Collaboration Lab.
Before I answer this question, I would like to thank again David Schramm, Mr. Grant, and Nina. Your great support, especially under the NIH NCI, and including the Solarz Memorial Foundation, give a lot of support. Indeed, we worked almost around 5 years. It's a long work, and a lot of the friendship. Just echoed David Shump. David, you talk about the trust, a lot of trust. The mutual trust is an important fundamentals and the pillar to support us. I thanks again, and also I thank the whole audience to support today's international collaborative conference, and we share a lot our contribution from the basic clinical translation medicine and the bridge together. I sincerely appreciate everybody's contribution. And then about the aspect, the future collaborative cooperation between the NIH Foundation, TVGH, and the NYCU. I think it's a very good platform to bring each others together, and also can get us strengthened from different sides, not only in Taiwan, the US, but also we know lung cancer is now the #1 mortality in Taiwan, and increase the mortality rate in the whole of Asia. Indeed, it's a global strengthening to everyone or every countries in the world. So that's it's a spotlight. We can collaboration and then the coherence of the, especially from the NIH, NCI, David Shump, your excellent clinical and the translational medicine and the basic the together's contribution. This delivery can give us a very, very successful platform and the model and running in the not only in the NCI, in US, in North American, and the lots can bring a successful model to Asian, to Taiwan. So that's what we so appreciate. And we want to use this multilateral collaboration. Of course, we can deliver the multi-function contribution and the multi-function efforts in the futures. And not only from a Caucasian, from US experience, but also combine it in Taiwan, in Asia, treated lung cancer, the different clinical experience. That's what would give the cancers, especially like the #1 life-threatening cancers in Taiwan, also in the world, in the global. We know a lot new advanced clinical trial and the new develop the drugs like the 3rd generation, maybe 4th generation TKI for the mutated the lung cancer, adenocarcinoma, or either others, including squamous lung carcinoma in Asian is also increased. So I think the more effort and the mode, not only from the clinical, not only from the Western, not only from the Taiwan, Asia, but if we can bring together, that we can get a very successful platform. Indeed, it's really a shortcut, and it's already a successful road made to conquer life-threatening cancers, including lung cancer. So I thank again everyone's contribution, and we want to share this experience at the platform in Taiwan, not only in the Taipei, we deliver it to whole Taiwan and the Asians. Thank you so much again. Thank you.
This is Sean on behalf of National Jiao Yang University. I'm the vice president. I have a question for the Dr. David Trump. As I'm not an expert in this area, so my question is regarding to the public point of view. So what would be the hope? What hope would be the hope or the promise for the patient who's suffering with the lung cancer or esophagus cancer or even the cancer on the thoracic side? What would be the hope for them? I mean, that's my question. I mean, my interesting question. David and Dr. David, yeah. Would you mind respond my question?
Well, I think that some of the talks actually really, hit on some of the major problems and some of the promise that we now hold with through using collaborative research to move forward. Certainly, we need to prevent lung cancer. We need to prevent esophageal cancer and other malignancies that are associated with known environmental exposures, such as cigarettes, alcohol, and asbestos in the case of mesothelioma and so forth. But we also need to be much better at treating advanced disease because the reality is that most patients are presenting with advanced disease. So I think that some of the work that Dr. Yang has presented today really is starting to chip away at that rock of metastatic disease in finding potential avenues to prevent metastatic disease or to treat metastatic disease and much more effectively and in a more targeted manner than chemotherapy has done traditionally. Certainly some of the emphasis that we have placed on trying to look at resistance mechanisms to immunotherapeutic regimens is also very germane because the fact of the matter is that although many patients receive immune checkpoint inhibitor therapy, relatively few actually benefit from it long term. And so that is an area where if we make any progress at all, it will have significant impact in terms of morbidity and mortality from cancer. And last thing, I think that we have still yet to fully realize the potential of stem cell biology in elucidating fundamental mechanisms that drive cancer formation and cancer metastasis. So I hope through this collaborative effort, with our focus being to some extent on stem cell biology and modeling, that we may be able to identify new targets that will be much more effective in terms of treatment of cancer in the future. Thank you for the question.
Thank you, David.
Thank you very much. The symposium has now come to the very end. I hope all of us will be benefited and inspired by these outstanding presentations. Wish you have a nice day and a nice night. Thank you.